Hello everyone and welcome to this remote content on Raman microscopy as a part of your multimodal modes of microscopy course. My name is Giuditta. I'm one of the lecturers of Maastricht University. In particular, I teach at the Faculty of Science and Engineer, and I'm a lecturer in chemistry actually. And if you actually check out what I did with my life, fundamentally I'm a materials chemist. I traveled a bit all over Europe before I landed in Maastricht in 2019. And you can see that my curriculum is not the one of a microscopist. A lot of you might be actually employed into some sort of biotech and stuff like that field. Fundamentally, microscopy is not something that I do with my life. I do other stuff, but a lot of the things that you do with microscopy come down to a sort of characterization method, and this is how I ended up looped in this course, because fundamentally Mark wanted additional information on your sample that you can actually get in something that is a combination with imaging. So when we say that we want to go beyond imaging, there is a reason for this in a recurring question. Like, you're looking at something, you can see something, but the question that comes with what you see is, how can I make sure that my scientific assumptions come in together with the sample? So I prepare the sample in a certain way. I'm expecting my even in vivo experiment to carry out in a certain way. How can I know for sure that I'm looking at something in particular? Usually common answers come into destroy your sample, analyze it in different way with a sequence of analytical techniques like mass spectrometry, chromatography, things like that. Or it comes with staining, which is very common. So you make sure that you have a material that will interact more with something that you're looking at specifically on your sample. So the surface under microscopy will look differently. But there is also a possibility for a slightly more niche answer, which is make sure that your microscope can act as a microscope, so as a tool of imaging for you and your eyes, but it will also come with additional information coming from your sample under the same condition. And this is exactly what Raman combined with microscopy can effectively do. So, what is Raman? It's a very chemistry thing at the end of the day. Some physicists actually use it, but if you're not a chemist or a physicist as a background, you might have not heard of Raman in your life. Raman, at its core, it's a spectroscopic method. So when we talk about a spectroscopic method, it's a general label for anything for which you have a source with a series of possible energies. The radiation that comes through from the source, it's your sample. Your sample will have some sort of response to this energy input, and your detector will be able to collect it. Now, this as a concept is very fundamental. Like, most matter will have some interaction with some sort of radiation. So in particular, Raman is a subset of this possibility of spectroscopy. And it's based on the Raman effect. Now, the Raman effect was first reported in Nature in 1928. The paper is very, very short and on point, and is named after this legend of an Indian scientist, Raman, who discovered this business with an equipment that, fundamentally speaking, we should be very impressed with, because this was not an easy task, and even less easy with equipment that back in the days was not uniform. So equipment that we would have in the Western world was not equivalently available in India, and yet Raman managed to record this effect in an incredibly systematic way. And fun fact, I was told reliably by my inorganic chemistry professors that there is like this running joke in the community that reminds everyone of the fact that Raman in 1930 have booked his trip to Stockholm to collect the Nobel Prize for his discovery before the Nobel Prizes were actually announced. So this was the level of confidence this man had in his work. But he was also right, because he did win the Nobel Prize back in 1930. And if you are interested in history, I would recommend checking out the history of Raman, because he's an incredibly prominent Indian scientist, and he was a legend of the Indian Scientific Academy. So he did a lot of stuff apart from discovering the Raman effect. And what is the Raman effect? The Raman effect is an inelastic scattering of visible light. So what do we mean with inelastic scattering? We mean that we bring radiation in, and usually a lot of radiation passes through without a change in energy. It often passes through with a change in direction even while keeping the energy, and that's called elastic scattering. In inelastic scattering, the photons that are changing the direction are also losing or gaining energy in the process. So, for instance, the fundamental experiment that led to the discovery of Raman was the fact that you could use sunlight, which is inherently a white light, a series of possible wavelengths, filter out every wavelength that was not the violet wavelength, scatter it on a liquid, so make the radiation hit the liquid and collect anything that came out. Most of the scattering will actually come out unchanged, because elastic scattering is very prominent, so you collect the same energy out as the one that you inputted in, but there is a portion of the scattered radiation that comes out at a different energy level. That different energy is actually the Raman scattered light. So if you were to then filter off, once again, so here if you filter off the violet, you know that you are not going to collect anything that was elastically scattered, but you're going to pass through all the radiation that had inelastic scattering. Now, one of the reasons why this was impressive when Raman did it is that inelastic scattering in the Raman way is incredibly weak. We are talking about one photon every 10 to the 6, 10 to the 10 incident photons. So this is an example of painstaking scientific commitment in trying to get your signal to noise to actually uniform to a reliable and recordable signal. This was made remarkably easy from the 1960s onwards, when you didn't have to use the sunlight, you can immediately shine a laser radiation. So incredible flux of photons, already monochromated. You don't lose anything in this mixture of filters. You are sure that you can actually irradiate your sample significantly, and that makes the Raman scattering come up. So if we were to look at this from a point of view of energy level, please don't be scared. I promise it's nothing scary. But energetically speaking, molecules tends to stay at a series of ground state energy levels. So every molecule that you have around you at normal conditions, which we are at the moment, has a stable ground state. Of course, if you were to cool or heat the sample extremely, this will change, but in our normal condition we can identify a ground state of the molecules. Now when we do a Rayleigh scattering, we are not absorbing the light, because there will be energy levels way above. This is quantum mechanics stuff, please do not block the video. there are energy levels above, if you manage to transition to the energy levels above, so these ones that you see here labeled as S1, you actually have basically a photon transition, so you excite the whole molecule to a higher energy state. But if you stay below, then the molecule is sort of like just sinking with the radiation, not enough to jump through. And in the Rayleigh scattering, you just bump the energy up. This is sort of like what we call a virtual level. It's not real, so you're not exciting your molecule. When you drop down with the same radiation. But within the ground state, you also have a series of vibrational modes. So these are things for which your molecule is not getting to a completely excited state, but is responding to this flux of energy with some vibration. And this is the thing that gives you spontaneous Raman effects. So you either could start from the basic vibrational level, go to this virtual state, come down, but you come down and your molecule is vibrationally active. So you come down with this difference, which is the vibrational energy of the molecule. Or you can go in anti-strokes. So your molecule, by chance, is already partially vibrationally active. And when it gets radiation in and spits it out with this inelastic interaction, it actually relaxes immediately to the ground vibrational state. So there is this additional vibrational state that is added to the energy. So in this case, the energy of the output is higher than the one that you put in. In this case, the energy that you collect on the outside is actually lower than the one that you put on the inside. There is also this possibility for which is a bit more complicated, in which you apply the same concept, but you bring the radiation you bring a radiation into the sample that can carry up almost at the excited level. So that brings the states in a sort of resonance state, so this effect will be much stronger, much more interesting, and it could highlight stuff that you wouldn't be able to see with these two methods, but this really requires some tricks, and we're not going to go into that. And how do we actually report this thing? So this thing always comes with this sort of shift. So you know what you put in. You want to know what are you collecting on the outside. So you could convert the energy of the photon that you're putting in into wave number, so scale it with the speed of light, fundamentally. And usually in Raman, to make sure that you are independent of, your results can be reported independently of the radiation that you put in, you tend to give the shift as the deviation from the initial. So you collect the final energy and then you focus on the shift from the initial energy. So what do we mean when we say you get to these vibrational excitation states? We mean that molecules can actually, they have their ensembles of bonds, and they can start vibrating on the bonds. So usually you have an equilibrium position in which everything should be set in the way it's set. But if there is some sort of excitation of the molecule, the easiest thing to do, the one that requires the least energy, is actually make the bonds vibrate and vibrate around the steady position. So it's not a huge vibration, but it's enough to sort of like dissipate or absorb energy. Now, you'll have to trust me on the fact that not all of these things can be activated equally, and especially not all of these things are active in the same way in every molecule. This gives you a significant amount of power, because fundamentally this type of interaction, if you ever done an infrared, this is almost a similar business, but infrared is at a completely different energy scale. So the two techniques are not overlapping with each other. They are usually regarded as complementary because there are certain signals that you can see in infrared. For instance, here you can see how do you see the transmittance of infrared with CO2 versus how can you see the absorption at a certain shift for Raman. So these things are on the same scale, and you can see that this signal is actually not here, but it appears if you go in Raman. So why is this interesting? Because the Raman signal in itself is a signature. So the molecule, as it goes by itself, will be able to show you something that is very related and peculiar to it. And this here is showing you exactly what we were commenting on, the fact that you don't want to report the final energy, because the final energy is fine as something to report, but it's variable depending on the input of the radiation. Now these two possible lasers are very, very common because it's easy to have the green laser and the red laser. But if your molecule is active with both of them, you might want to measure it with both lasers. And in theory, regardless if the molecule is still active, regardless of what you input, you should still get the same trace of what is the shift. So this deviation will always be the same. So it is better for you to report the shift instead of the final energy, because that will be easy to tabulate, and no one will have to tell you every time, wait a second, I was actually using this radiation, or actually we have a bespoke laser in our lab and we are completely here in the middle. It doesn't matter. It's normalized for the incoming radiation. And whatever comes here is actually structured in a way that is responding to whatever you have in a certain molecule that can actually vibrate and get like partially like responsive. I wouldn't say excited, because excited is that thing up there and it's incorrect, but something that can be responsive to your incident light. So do you see the peaks or not? That's something that depends on how polarizable your molecule is. So it's basically how your molecule feels this electromagnetic radiation passing through. This is related to the symmetry of the molecule. You have to trust me on this. If you have specific question, I can actually unpack the business from a symmetry point of view. But the fact that this relates to the symmetry means that you don't have to worry too much, because probably there might be some crazy chemist around the world like me that actually did the modeling of the specific molecule that you're using and might tell you immediately, this is active, this is active, this is active, and at this frequency. So how many peaks will actually depend on how big is your compound, how many degrees of freedom does it have, so how many variability of the configuration of this molecule you can have. And at what frequency depends on the constituents, but given how these things change with each other, a Raman spectra can be predicted for a given molecule. It's not even too difficult if you have the right packages for chemists. It could be a bit more challenging for certain stuff, but also there is a lot of experimentally reported data. And usually you tend to collect strokes Raman, because the strokes Raman is over here, and the strokes Raman is the one that loses energy in comparison to the one that you're putting in. This is stronger. You can see here the ratio between the intensity of these two peaks, and given that Raman is already kind of weak, there are people that go into anti-strokes Raman, but you might find most of the instruments as working in strokes. So the incident radiation and the absorption spectrum usually goes like this. The advantage of the anti-strokes is that you could fall into a dropping absorption spectrum, so we could show you stuff that here will get swallowed by the actual absorption. So the absorption is actually exciting everything, so it's not the one that you want. So you tend to collect the strokes Raman, and most of the spectra that I'm going to show you are actually strokes Raman spectra. Now, a thing that is very nice as materials chemist is that the actual crystalline materials have a lot of Raman signal, because a crystalline material has a bunch of atoms all arranged symmetrically in space. It's a lot of them, and they all follow the same arrangement. This means that you could track what happens to something that you know is going into, for instance, a mineral phase into your material with Raman. Here is, for instance, as a function of time at a certain temperature, and you can see that when you actually grow the anatase, which is a titanium dioxide mineral, the Raman signal skyrockets. And here you can see this series of labels, and the labels are actually symmetry labels that are used among chemists. You don't have to worry too much about them. If you were to look for anatase Raman spectra on the internet, it will pop up, and you will just know that this is exactly what you're ordering. So, of course, you still need to have Raman active modes. There are crystalline material that are Raman inactive because of the symmetry. That happens, but if the arrangement of the atoms in your crystal allow it, your crystal will have a Raman signal. Another reason why Raman is used specifically is to be able to measure surfaces and also solutions. So you don't have to have it on a solid. Solutions are Raman active. So you could have if you have like Raman instruments with particular sensitivities, you can see nanoparticles growing onto a surface, or functionalized surface getting modified, or something happening into a solution. Raman will track the appearance of this signal, because it's seeing a chemical signature that is either appearing or changing or stuff like that. Warning for this, these images, for instance, are for surface enhanced Raman scattering. So this might actually need more complicated Raman setup, but if you end up needing this exactly, there will be people, especially in chemistry department, that might be able to help you with this. But I feel like the most interesting part for most of the attendees of this workshop is actually the Raman of biological molecules. So biological molecules are carbon-based, but Raman doesn't care that you have very light atoms. What you care about is the fact that the bonds of these atoms actually tend to have a polarizability that is frankly fairly high. Like, it's very rare that if you stretch a biological molecule around, the arrangement of the electrons in the bonds remains exactly the same, exactly symmetrical, which means that fundamentally biological molecules are Raman active. And especially the macromolecular biological molecule will have a repeat arrangement that will actually create a series of features that are kind of unique. Because if it were to be all the molecules, all disordered, just a series of CH, CCC, CO bonds, it will be basically impossible to distinguish them. But all biological molecules tend to have this higher hierarchy, and the higher hierarchy will also come into how sensitive they are into the radiation, and on which levels do you exactly fall when you do this inelastic scattering. So when you see here the spectra from a water solution of biological molecule, if you look at the sequence of peaks, even without worrying about what are these peaks exactly, you could see that even DNA and RNA, you could argue they are distinguishable from each other. Look at this signature over here. Here you have this sequence of the triple peak, but the triple peak as it is here, with this distance between these two peaks, is completely different from this double peak that you see of the RNA. And on the other hand, like other stuff like lipids will be distinguishable because they have peaks at different intensity. The shift in wave number does not overlap. So it can really be a quick tool to just tell you where are we and what am I seeing if you do it properly. You need to remember that depending on the arrangement, here things could change. So you can, Raman definitely has the power to track different molecules and different chemical compounds as a fingerprint around. Remember that if you have something very specifically ordered, you need to be careful to do like enough measurements so that the orientational disorder doesn't kill your Raman signal. You can see here basically same molecule in one direction or the other direction. This is a fatty acid. If you end up having them oriented against where is the Raman source coming into, the overall spectra is still the same. So if you're just interested in the overall fingerprint, it's not a problem. But if you have a strong orientation, it could affect the intensity of your Raman signal. And even among things that are really similar to each other, like phospholipid against cholesteric acid against triglycerides, you can actually see the difference between them. So even with molecules that you can see the spectra are similar, not so similar that if you are resilient enough, your Raman spectra will not show you things. Now, of course, these are biological molecules, so all the samples that were put in the Raman were sort of like purified to start to show only that. In a biological sample, you will have a spectra that is inevitably way more complex, because if you have like a whole section of a live experiment that you just did a cross section of and you want your cryoslice to go into the Raman, then you are going to see all of them depending on where you are in your section, what is your tissue made of. You cannot say, oh, this tissue is just fat. It's not going to be just fat. There are going to be some proteins. You're going to see maybe some DNA if you are focused on a cell or something like that. So you can track what is the most interesting aspect of a certain biological molecule. You have to expect that unlike the previous one for pure molecule, you're going to have a lot of noise. Luckily, there are people that spend their life on this thing, so if you look nicely enough in the literature, you are going to be able to keep track of what is reported to be a frequency in wave number that you should actually look for if you're looking for something. And you can see there is some redundancy. So for instance, if you only see these two but you don't see this other one, then you might have to be a bit careful, or be careful that things are not overlapping. But this was a lot of spectroscopy, right? And I promised you that this was going to be a mix between microscopy and spectroscopy. So what is the point of crossing over Raman in a microscope, and why can they be this crossed over this easily? This is because Raman is a technique based on visible light. So whereas, as I told you, it's complementary to infrared, but infrared, you can only see it as a fake contrast. Whereas, so an infrared microscope is not going to be able to show you an image, because when you look into the microscope, you do not see the image. It needs to be processed afterwards. But a Raman, as a signal, is based on visible light. So your microscope that you're using to do imaging of the tissue, if it's properly set up, with this double source and the detector could actually lend you a hand and do microscopy, because it's using the same type of radiation. The lenses will look the same. Of course, whereas you use usually white light to be able to image a whole sample, so you tend to just open up the lamp and do everything, you will have to have a laser beam to be able to do Raman, and the laser beam is monochromatic, but it's much easier to match the optics if you're using the same range of radiation. So the optics for a microscope are going to be more similar to the optics for a visible laser than they are for a laser that works in infrared, because the distances are different, the grating are different. So usually what you do is you want to be able to have sort of like a filter that comes through, so you can stop things and split them off. So when your microscope is working as a microscope, it cannot work as a Raman. If you are looking at something visible, then the laser cannot work. You need to shut off the light and start to irradiate the sample, but you can have a control that does it for you. I will show you in the practical section. And what you want to make sure of is that then you collect whatever radiation comes out, and the radiation gets split into the grating and gets to the detector, so the detector is able to really differentiate through all the energies that you actually spit out. So this is the classical setup of a Raman microscope. So this is the usual microscope, but there is a whole set that has an incoming laser. The laser gets in, and the radiation that comes out, you want it not overlapping with the line that brings the laser in, but you want it coming out, filtered, focused, and then grated off, so you can differentiate all the energies this is composed of, and then lands into a CCD camera. With the fact that you can combine the microscopy and the Raman, when you have an instrument that is properly set up with a proper computer, you can combine what you see as an optical image and make it track the Raman signal. So you can just say, okay, now go into this whole thing and go step by step and collect me the Raman signal. Especially you could focus it at a specific energy. So for instance, these maps were tracking a specific band at 1004 centimeters to the minus 1. So in these images you only see intensity when the band is popping up. If there is nothing there, then you will not see anything. And you can see here that this went all the way down to 0.25 micron step size, so it can be pretty precise. So here you can image off the whole thing, so you can reproduce the image, but the only thing you have to be mindful of is that if this thing was taken at a bigger scale in light, the light is fine, the light can still focus, but to focus a laser you need to stay on a single spot. So if you look at the formula as I put them, you cannot do imaging with the laser, because the laser doesn't have the aperture of the beam to actually take up the whole image. So you can focus this beam to allow you to do Raman, but it will have to focus on a spot that is around 1 micron, if not smaller, depending on how good are your optics, fundamentally speaking. So when you do Raman, when you do the imaging, you need to keep track of the position you are at, because to be able to use the laser, you need to stay at a different magnification, a magnification that will allow to focus the laser beam. With a 5-pair objective, you are able to see the image. It's perfectly fine for a broad visible light, but the laser will not go into focus. These lenses are too big, so you need to be with an objective that allows for this trick of the focusing of the beam. That said, that can be done because if you are able to track the position, it's feasible. And you might want, since we were seeing before that you have different type of bands for different things, you could ask, since you are collecting every time the whole spectrum, if you give it the proper range where you want to collect, you might as well ask the processing software to just track different things. So I took this thing from this reference, and you can see here from the same sample you see different bands at different positions. So overall you know more or less what you're looking for, but you can see that the signal of certain stuff does not appear uniformly through all the samples. So you can see this stuff that is focused here, this thing that is instead more on the side, here there is almost nothing, here you have like some discontinuities, here you can really track the whole business with layers. So when you mix the two of them, you actually end up with a lot of power if you do this properly, because you first reconstruct the image and then you gradually map everything. So this could actually be an interesting technique, because then it's quantitative even. You could see like different intensities. Of course, the quantification could be slightly delicate, and it's also molecule specific. So no one is going to ask you, why are you telling me that this signal is from this molecule? If you did your research properly, then you know that is reported to be that one, and you can even collect standards of whatever you're doing before. So you already have your own experimental standard data, and you can see how well do they match when you go in. So this turns an imaging technique into something that allows you to look at your sample, but it also allows you to map the chemistry of your sample on a very fundamental level. And this can be incredibly powerful, a bit delicate, but definitely worth the effort. So to summarize, Raman is a spectroscopic technique. You irradiate with laser invisible light, and whatever energy you get at the on the outside is a fingerprint of your molecule. A lot of molecules, even in biological systems, are active, and you could couple it with microscopy to make sure that whatever spot you are looking at in invisible light will be remapped with the laser of the Raman. And you might have noticed that in no point in this presentation I talked about labeling, because you don't have to label anything. The chemistry is doing the label by itself. A bit of warning: calculating the fingerprint can be done by first principles. You might get lucky and it's already done. If it's not done, might be because it's challenging. Maybe your molecule is incredibly big, you want to track a whole protein, that things are complicated, and you might want reference samples or a lot of reference literature to make your life easier, so you don't have to reinvent the wheel every time you go to a microscope. A laser is incredibly powerful. This is a point of warning for when you're using biological sample under a Raman. Lasers are strong. This thing can burn your sample through, especially if you collect for very long at very high intensity. So some tweaking is required to make sure that you acquire the most of the data in the most delicate way with the best signal to noise. And a thing that you might be already aware of, since you might have never seen a Raman microscope before, is that they are not widespread, so you might actually need a bit of support, and it might be worth actually checking out with your chemistry departments, or if you have any industrial partner that you know works in sort of like materials or chemistry or surfaces, because they might have a Raman microscope and they might have someone willing to support you in the track of learning how to use this microphone, microscope. and I will show you how it works with the microscope that we have here at Maastricht University. So the next video is all going to be about showing you a real sequence of acquisitions in a very quick way. And in the meantime, I'll thank you for sticking around for this video. Please note down any questions, because I aim to be able to do a Q&A session. And if you have anything that you want to make me aware of, please drop me an email. I'm always available. And you can check some of the literature that I based my presentation off, which has strong relevance on biological systems, because I feel like they might be the most needed in this case. Thank you for tuning in on the video. I will see you in the next video, where we discuss actual practical stuff, and possibly during the live Q&A.